I know. So my name is Ryan O'Connor. I'm part of I'm working with RDA, the RDA Association, as part of which is essentially RDA Europe, as part of the RDA Tiger project. So I'm not a co-chair of the FAIRification of Genomic Annotations working group. I'm helping out. I'm kind of one of the facilitators from the Tiger project. So I'm helping out, kind of supporting this group. So welcome to the session. This is the first session of the working group since it's been sort of officially endorsed by RDA, titled Metadata Harmonization at Scale. So as I said here, I'll just put into the chat the link to the notes. If you just go on to the next slide, please, Swino. Yeah, so as you might have got, you're probably all aware now, and you probably heard when you joined the session. The session is being recorded. As I said, I've put a link into the chat for the collaborative notes. If you could all just if you could just add your name to the top of the attendees, to the table at the top of that document, the attendees list, that would be much appreciated. And yeah, also with just a brief bit of housekeeping. If you just keep your mics on mute for the opening parts of the session, we will there will be spaces for Q&A and a kind of larger space for discussion a little bit later. Next slide. Yeah, so just to briefly run through the agenda, and again, this agenda is copy and pasted into the collaborative notes just so you can refer back to it later if you need. So yeah, we'll just run through the welcome, and I'll hand over to Sveinung, who is one of the co-chairs of the working group, to give a brief introduction to genomic annotations now, and we'll then move on to a couple of lightning talks on use cases and previous work. So that'll be approximately about half an hour. So four of these five lightning talks are recorded, so I'll be sharing my screen at that stage and sharing the recordings with you, and sharing the recordings on my screen. So these will be from the additional three co-chairs of the as well as Sveinung, the additional three co-chairs of the working group. So those are Anna Bernasconi, Nathan Sheffield, and Adam Wright, and we have an additional presentation from Thomas Brown. And they will introduce themselves in their recordings. We'll then have a brief section after those lightning talks for any questions that you might have. And I'll hand back over to Sveinung, who will be taking us through a kind of overview of the working group's work plan. And then we'll be opening the discussion up to kind of look more directly at the kind of challenges within the use cases, which will be described, the tasks that the working group has outlined in its work plan, and kind of possible intersections with other RDA groups and efforts elsewhere before we wrap up towards the end. So with this, I will hand over to Sveinung. So I'll hand over to you, Sveinung, and the floor is yours.
Thank you. So you can hear me fine, I hope.
Yep.
Yes. So in Norway now it's past midnight, so please apologize if I'm a bit late or slow when talking here, but I'll do my best. So my name is Sveinung Gundersen. I work in Elixir Norway for the University of Oslo. So I'm going to say a few words about genomic annotations just to sort of set the stage for the lightning talks that will come afterwards. And sort of start a bit at the very beginning here for those who don't really know life science and bioinformatics. So the main concept here, another word for the annotations, is genomic tracks. And I'll go a bit through what that means. So this is a unified model for data analysis. So starting at the very left here, you can say that a genome, which is the totality of the DNA in an individual, can be expressed as a sequence of letters, one for each of the 4 nucleotides. And there's an example here. So in order to make any sense of this sequence, you need to add other information to this. You need to annotate it, figure out what are different elements, where are functional processes happening, and so on. One basic example of this is to position the genes, figure out which part of the DNA that's translated into proteins. This is basically what a genomic track is. So we have a series of these related data elements positioned along a reference genome sequence. So that is what we're working with here. This is the data that we are going to verify. So this type of data is used, was designed basically for use in type of analysis software called genome browsers, which you can see an example of here to the left, which is the UCSC genome browser, but also used in a ton of different analysis tools. And even though this is a sort of very visual concept, and a track is something that you can sort of horizontal line here with some data. This data model is also important for non-visual analysis. So there's some examples here on the left, on the right. And there are lots of different data types that can be expressed in this way. And I mentioned a few here. I will not go through this list in detail, but everything from the genes, as I mentioned, to different types of regulatory elements. And then importantly, there's a ton of different experiments, lab experiments that can be visualized, where the outputs can be provided in this way. And then you can compare those experiments with those results with other knowledge that you might have. So this has been talking about genomic tracks, and then there's a similar term called genome annotation. And what does that mean? Well, basically, it's very overlapping, but genome annotation is typically the initial characterization of a genome. So when you have sequenced a species or a human, then you start a pipeline where you find the different important regions, like the genes, like regulatory elements, and so on. So basically, this is the original type of track. But I'll come back to why there are different terms on this concept. Yeah, so starting to envision and figure out this working group, we were thinking a lot of different names. What should we actually call this? Because we have these tracks, we have genome annotations, and lots of different words that actually is used in literature, and there's no uniform names for this, basically. We came up with the term genomic annotation, which is slightly different from genome annotation, just to make sure that it's not exactly the same. It's a bit meant to be a bit broader. And there's some support in literature for this. And the reason why there's a bit of difference in nomenclature here is that there is research fields also a bit split. So on the one side, there's the functional genomics and epigenomics field, which is historically been mostly on human biomedical research and also all the other model organisms. And then you typically have a coordinate system that is the same, and then you put different types of tracks on that and compare them. While the actual genomes and annotations, the reference genomes, are typically generated centrally by large consortia or repositories. While on the other hand, you have the biodiversity field and the comparative genomics field, where there's the main focus is comparing different species in the tree of life, and then you have also different coordinate systems, because the genome sequence define the coordinate system here. And then also lots of different projects that are now assembling and annotating genomes. So there are different technical challenges in these two fields, but in the main concepts, they are working with the same things, and genome annotations, which is and tracks are basically the same concept. So what is the basis for this working group? There has been a ton of different large consortia that have created data, raw data, and then this type of track data, which is a more of a summary form of data, costing billions to produce in total. And all of these different projects, and most of the projects, end up with a data portal with metadata connected to, but they are largely incompatible. And also the APIs to use them are different. And in many smaller projects, there's little metadata. So there's been a need for a harmonized metadata model for many years. And then on the biodiversity side, as I mentioned, there's lots of different projects, most of which are now under the umbrella of the Earth Biogenome Project. And the goal here in total is basically to sequence millions of new genomes, sequence as much as possible of all the different species that we have. Okay, so back to the aim of the working group. Can we harmonize and verify genomic annotation metadata across all of these projects and beyond? That's the main goal here. And also adding to that is the question of whether we can build a simple community-oriented infrastructure for discovery and reuse of genomic annotations. And this is what we will try to do in this working group called FAIRification of Genomic Annotations. Okay, so that was my introduction, and now on to the lightning talks. So first there's a lightning talk by Anna Bernasconi, who's the co-chair.
Good. Thanks, Final. Yeah, so I will share my screen now. So I have that on screen. 1 second. Share my desktop. And I'll switch to Anne's presentation.
Dear all, I'm presenting building integrated and fair repositories for genomic data. Mainly, I'm going to present the results of the Data-Driven Genomic Computing ERC Advanced Grant led by Professor Stefano Ceri, where I worked for the last 5 years. The main outcomes of the project were 3 abstractions: genomic data model, genomic query language, and the genomic conceptual model. The genomic data model is a model where files of genomic regions or tracks are called samples and
Yeah, something happened with the audio.
we can represent multiple data sets of genomic regions or tracks as tracks as you see them here: promoters, peaks of chip sequencing or mutation DNA sequencing.
Yes,
First,
it's working. Can you rewind a bit?
The genes to extract the promoters region data file where.
Apologies, I muted my microphone. I didn't realize it would mute the sound as well. Is this about as far back?
Yeah, that's fine. Fine, I think, yeah.
Apologies.
Of genomic regions or tracks are called samples and divided by dataset. Each sample has a region data file where regions are chromosome number, start and position, and a strand with their properties. They are attached to a metadata file, which is a list of key value pairs. Thanks to the representation in genomic data model, we can use the geometric query language system. In this system, we can represent multiple data sets of genomic regions or tracks as tracks as you see them here: promoters, peaks of chip sequencing or mutation DNA sequencing, for example. Here we project the genes to extract the promoters regions, then we do a cover operation to only confirm those tracks that are confirmed in at least 2. We do a distal join with peaks and promoters. We do a map with the mutations of breast cancer, and then we select only those promoters that are confirmed in the region position of the mutations. We also have the genomic conceptual model that allows us to add a layer of semantics to the very simple representation of key value pairs of metadata. Here we have that the central entity is the item, also the genomic data model sample, and it is described using 4 semantic directions: the biological information, donor, by sample replicate, the management view, case study and project, the technology, about the experiment type, and the data set for the extraction view. Thanks to this representation, we were able to build a big FAIR repository where we downloaded the data, transformed and cleaned information, mapped the metadata information into a relational database design on top of our conceptual model, and then enriched the information by normalizing it with biomedical ontologies, and thereby improving the interoperability and reusability of our data. As a bird's eye view of the whole system, we have that our user do not have to query separated interfaces, but can use one single entrance point called GenoServe, which is a search engine on top of the Metabase repository. It allows you to build complex queries by combining different attributes and values conjunctively and disjunctively, respectively. Once the query is composed, a list of samples is retrieved, and this list of samples can be directly sent to GenQuel, the system that I described before, that allows you to build complex biological queries and resolve complex problems. We also have a federated implementation of this. I would like also to introduce the very latest project that we're working on, the Better Real World Health Data Distributed Analytics Research Platform. It's a Horizon Europe project, and we are partnering with other universities, research hospitals, and companies. Here, the goal is to build a decentralized infrastructure to empower healthcare professionals to exploit large sets of multi-source health data. Here we have clinical health data, but also genomic data, including VCF files, mutation data, and images of retina. The goal is to have a secure, cost-effective, across national borders, complying with GDPR platform. I'm working in the Politecnico di Milano unit, and here we are responsible with the health dataset verification and pre-processing actions. How does the platform work? Basically, we get the raw health data and we build pipelines to verify this data. So we will build for each hospital a FAIR database that has all the information in the same format, same semantics, same understanding, so that the personal health trained that works in the federated learning process can access this data in the same way. Thank you very much. Here I leave you the bibliography, and I close here.
Great. Thanks, Anna. Obviously not here to take our thanks, but apologies that I didn't realize that muting my own microphone would cancel the sound. So next up is Sveinung, who will be giving a short presentation. So I'll hand the floor back to you, Sveinung.
Thank you, Ryan. Yeah, so here it is. Yeah, so as often happens, there are different initiatives trying to do similar things, and the project that Anna just described was happening in parallel with the FAIRtracks project, which I am now going to say a few words about, without really knowing that we were aiming at more or less the same things. But this was then a project that started a few years ago. So the background for this is a tool called Genomic Hyperbrowser, which I have been developing, a part of the development team since 2008 or something, and which is a tool for statistical analysis of tracks or annotations. And what we discovered there is that we needed a way to import data with metadata And we tried various ways of doing that, different import tools. And in the end, we ended up with a idea that we needed to create more of a harmonized standard for metadata and solution for that. And that was the background for the FAIRtracks project, which started as a implementation study in Elixir, as a collaboration between Elixir Norway, Elixir Spain, and the EMBL EBI. And then the results were published 2021 with some recommendations trying to figure out how to best verify genomic tracks and add metadata. And this output was also selected as an Elixir recommended interoperability resource. So this was sort of the basis of this working group, and we found out that in order to get this to scale and become a potential global solution, we needed to include a larger community, and then that was the start of the RDA working group, and we got in contact with the RDA Tiger proposal, and have now been working for some years to prepare this working group, which is now starting. So a few words with what we the results from the FAIRtracks project. As I mentioned, we published recommendations looking at the different principles and trying to pinpoint how those could be adopted for track metadata. And in the same way as Anna showed, we also designed a prototype metadata model using JSON schema. So this is not meant to have all possible metadata for an experiment and so on, but a minimal set which is useful for discovery and reusability. Different interoperability schemes. I will not go into detail with all of this, but basically these 5 object types are very similar to what Anna also came up with, so that's a good background here. We also designed different services connected to this schema with validation and augmentation of metadata, and also a search service that could then search through the harmonized metadata, also connected to a Galaxy platform. Yeah, so this, I will not go into detail with all of this, but we have been thinking about how this could be expanded to a larger data lifecycle, figuring out how this could be done in the context of existing metadata and data preparation and publishing routines. So this is an overview of what we're thinking, and I can go through that in detail later. We also developed a website which is called fairtracks.net, which is designed to function as a community website. And there's lots of information here about tracks, genome browser, FAIR concepts, and all of the things that I've been talking about. Currently, it's focused on the services and outputs from this previous FAIRtracks project, but this can easily be transformed to function as a hub for the new RDA working group. So that is at least my aim. Also briefly going to mention another project called OmniPy, which is a Python library that we have been developing the last years in preparation for this working group and other projects. So basically this is for data wrangling and transformation of metadata. So there is a large need to get the existing metadata in the different, according to different models, and get them harmonized into this new metadata model that we are defining, and how to do that in a simple and scalable way. That's the problem that we're trying to look at here. So this is a way of using Python to specify data models and with powerful features for parsing, validation, conversion, and interactive tools, and also for orchestrating these transformation workflows into compute infrastructure. So this is the people that were involved in the FAIRtracks project. and the different funding for that. So that was my talk.
Thanks, Swino. So I'll take over again. It's just a few more quick lightning talks. So I will once again share my screen. So next up is a lightning talk from one of the other co-chairs. Again, this is a recorded lightning talk for one of the co-chairs of the working group, Nathan Sheffield. So I'll just press play on Nathan's recording now.
Hi, I'm Nathan Sheffield. I'm an associate professor at the University of Virginia. My lab studies human epigenomes, and we have a special interest in breaking down barriers to data access and use. Today, I want to describe a project we're working on that addresses some of these challenges with epigenome data access. The project is called BedBase. It's basically a database of bed files. So we're trying to catalog all the published experimental genomic region data sets that we can find, put them in a central location, and make the data more usable. So an important goal of BedBase is to make the data easier to find and easier to access. We're working on a web interface, command line client, as well as Python and R packages. So these different interfaces would allow you to retrieve, cache, and analyze the genomic region data in a variety of ways, interactively, as part of a tool if you're a tool developer, or in a reproducible script, whatever you need. So what's the problem that we're trying to solve? Well, as you probably know, human epigenome data has been exploding. There are now almost 100,000 bed files available on the Gene Expression Omnibus. So it's great that there's so much data available, but the problem is that it can be hard to find. Maybe even more importantly, it can be hard to access and use because the interfaces aren't really set up specifically for bed file data in terms of making them easy to use interactively. So to give you an idea of what I mean, here's a little video where I'm searching for some data. So I look for Ewing sarcoma, ChIP-seq data. This is a cancer that I'm interested in. If you search for this, you can find some data on GEO, and you get a list of possible results, and you can identify some ChIP-seq data that you're interested in. So this particular example has some ChIP-seq data, and the way you could find it, get access to it, the actual files themselves, you look down here in this custom menu, and here we have these narrow-peak files, and narrow-peak are files that are identifying genomic regions, similar to bed files. You could select a few files of interest and then download the data, and this is basically going to download an archive, a tar archive of those files, which you can then look into the archive and grab the gzipped narrow-peak files for the different samples of interest. So this is sort of fine, but it's really geared toward a human, less easy for a computer to do. Now, if you want to look at metadata, you have sort of a similar protocol where you're going to use this FTP server and use the FTP server to download a compressed soft file. Then you can open up the soft file, and at the end of the day, you're going to end up with metadata that looks something like this. So a lot of really useful information in here, but in a format that's a little bit difficult to parse and use in your downstream analysis. So it's great. GEO is great, and we use it all the time, and it's an excellent source of data. But it can be a little bit difficult to use and to get access to the data. So I'm going to show you a little bit of the alternative of what we're thinking with BedBase. So we're working on, first of all, a search interface that's going to use semantics to make it easier to find what you're looking for, but I won't talk much about that now. If you've got a specific bed file that you're looking for, you're going to get a view that looks like this, that gives a lot of information about that bed file immediately. Because BedBase is specific to genomic region data, we can build interfaces that are really geared toward this data type to show you things like the number of regions and what is the distribution, as well as a bunch of other at a glance information to help you know if this is going to be the file that you're interested in. In addition to this, everything's API oriented. So the point is that this is programmatically accessible. All the different statistics that we've computed about the data, as well as some analyses results. And you can also use the API to get the bed file itself, or a compressed version in a big bed file. In addition to this, there's we're making this open API compatible. So there's documentation for all the API endpoints. And so that's basically a brief overview of what BedBase is. So the goal here is to provide a really nice interface for this really important data type, so that tool developers and data analysts can make better use of the huge resources of genomic region data that already exist. So this is why I'm interested in our new Genomic Annotations RDA working group, in which we're going to be developing community standards around how the metadata for these files should look, and hopefully also get to do a little bit of work around how APIs like BedBase can communicate with one another to make it easier for all of us to use this really important data type. So thanks for listening. I hope you will give BedBase a try as we develop it, and feel free to drop me a line with any feedback or ideas. Thanks.
So thank you, Nathan, in absentia again. So I should mention as well, so we've two more recordings there. Next one by Adam Wright, who's one of the group co-chairs. And after the final one from Tom Brown. If you have any questions on any of these presentations, obviously the presenters aren't here, but either, I mean, Sveinung might be able to answer them. I certainly won't be able to answer any questions you have. But if you do have any questions, please feel free to post them in the chat, or even ask them, and we can kind of make the connection with the presenter, and hopefully they'll be able to kind of follow up on those in some capacity. So as I said, next short presentation from one of the other group co-chairs, Adam Wright. Apologies, there is a bit of an issue with the sound of this video. There is a bit of a kind of buzz in the background, so but it is legible. It is just a bit difficult to listen to, so apologies for that in advance. So I'll just hand off to Adam.
Hello, I'm Adam Wright. I'm a technical manager on the Reactome project. I'm also 1 day a week on the Alliance of Genomic Resources. I've been in the field for over a decade, and one of the side things that I like to do is work on coordinating metadata and metadata standards, and I co-chair on this working group. I'd like to talk to you today about two different topics that have to do with one another. JBrowse genome browser, and a reference genome header specification. And it will become clear, hopefully, as I go, why I'm presenting on these two things, and then I'll explain my proposal for what we could potentially do with this within this working group. So JBrowse itself is a fairly well-used, widely used genome browser that anybody can install. It's open source, and you can configure it to have your own tracks. You can pull in tracks from others, from their instances. You can have whatever reference genomes that you would like. You can have DNA or protein sequences. It has a lot of different visualizations. One of the things that I want to focus on with this is that when you click on the name of the track to the right, there's 3 dots there. You can click there and get more information on that track. So that's where you would get the metadata for the track, which is similar to what this working group is working on. So with all sorts of genome browsers is the intent for this working group, but I want to focus on JBrowse. I think that it's a good starting point for this sort of work. So another thing that I have published earlier this year with a group of collaborators is a file header specification for reference genome files. Normally, FASTA files don't have a file header. The format of ours is the same as YAML, and then but with a semicolon tilde after it, denoting that this is the header. Normally in the original FASTA specification, the semicolon denoted a comment, so we're taking advantage of the comment line. Not all tools will run with this, but it's a great way to put information in there, and we find it to be minimally invasive, being that if you want to run this through a piece of software that can't ignore these comment lines, you could simply take out the header. We provided the tools for that, or you can do it whatever way you like. But having ever increasing number of reference genomes it is creating a need for having these sort of specifications. So here's the configuration for JBrowse that I talked about earlier, where you can, within the JSON file, you can specify for a track the metadata file that you're using. It will show up as text within the JBrowse interface. I'll show you that on the next slide. But for each track, you can specify metadata, and there is no standard right now, and the standards created, or the specifications created by this group, could be used and recommended to users to use, and tools can be made to help them make metadata for all of the different tracks that follow FAIR principles. So here's one of the places where this has already been taken on is within the Alliance of Genomic Resources. For each one of the species, there's something like 6 species now that have joined the project. They all have JBrowse instances, and a lot of the mods will have many JBrowse instances for each one of the reference genomes. And so within these JBrowse instances, we have added, well, Scott Kane in particular, that's managing that. He has been creating headers that follow our specification that show up within the browser. And yeah, this is all configured through that configuration file in the previous slide. Very simple, but gives the user a lot more information than just like the information above that provides the information on the based on the file name. And so yeah, what I'm proposing is that we create a tool, say in Python or something like that. We could agree upon that, that goes and validates the JSON configuration file for JBrowse to make sure that it follows in the specification that this group comes up with. And so information get pulled from somewhere to make sure that the information in the tracks are fair, or also just it could be a validation tool that it follows a certain set of rules that we come up with within the group that could come up with later, but it's a simple project that could have a reasonable impact. That's it for me. Thank you for listening. Bye.
Thank you, Adam. So I'll just see there's a message in the chat. Yeah, so the final of the quick lightning talks is from Thomas Brown from the Leibniz Institute of Zoo and Wildlife Research. So Thomas isn't a co-chair of the group, but I think he was invited to provide a quick lightning talk by Sveinung, as one of the group's use cases is on biodiversity. So we just wanted to get his thoughts on the possible impact of this group and the potential for it in his field. So I'll just press play on Tom's talk now.
Hello, my name's Tom Brown, and I'm the coordinator of the IT and Infrastructure Committee in ERGA, or the European Reference Genome Atlas, which is itself the European node of the Earth Biogenome Project. And I'm going to be giving you the biodiversity perspective on genome annotations and some of the roadblocks we're facing in terms of publishing, using, and reusing them across the tree of life, and how we've started the process of trying to make genome annotations a fairer process in terms of producing them, publishing, and reusing them in the community. So the EBP and a number of regional and other nodes have the ambitious aim of sequencing and producing reference quality genomes for all eukaryotes on Earth. And this has resulted in an explosion in the number of these assemblies being published, but the annotation of these genomes, and in particular the publishing of these annotations, is lagging behind. And I've shown this simply in this graph where I've plotted in black the number of cumulative reference genomes per year, which are deposited in GenBank, and below those in blue, which contain a RefSeq annotation, or in red, a user-submitted GenBank annotation. This really highlights that thanks to technological software and the sheer democratization advances of the process of reference genome generation, we have a huge number of genomes at use, but not many that have public annotations which can be freely exchanged via databases such as GenBank. And this is a huge issue because a large number of the downstream analyses which we want to perform rely on the annotation of a genome, and in particular, being able to use the same annotation for analyses in different research groups. So the reason why this is the case is complex, but one of the consistent issues we fall into is inconsistent file formats, and in particular, different databanks accepting different file formats. So ENA and NCBI both accept different formats themselves if one wants to deposit an annotation in their databank, either MBL or table format. And this itself can only be deposited by the assembly team. So both an annotation and the genome assembly can be packaged into an MBL or table format and submitted to ENA or NCBI. These file formats themselves are not generally used by researchers or software which they would use to analyze genomes. And in particular, if a third-party research group wants to publish an annotation, this cannot currently be done. And this is a huge roadblock in terms of sharing annotations once published with other researchers. So currently the state of play is that normally research groups will create ad hoc annotations, doing the best they can with methods normally shared in papers or briefly written in databanks. And this has created a large reusability problem among the field. However, we're hoping this will improve in the future as the community is starting conversations with both in ENA and NCBI to accept both a file format which is accepted by the field, for example, GFF3, and one that contains the necessary information in terms of understand what it contains and how it's been made and by whom. And hopefully, soon these databanks will accept such annotations in part of their submission process. Within ERGA, we're currently working in an EU Horizon project called Biodiversity Genomics Europe to produce and annotate around 500 reference genomes for species found in Europe. Not only are we able to publish these annotations, but we're also working to develop annotation workflows and benchmark them across as many species as possible in the tree of life. We're working closely with Ensemble to make sure these are of a high standard, but also Workflow Hub to be able to publish our workflows written in SnakeMake, Nextflow, or Galaxy in a format with full software and versions, and can be redeployed by anyone with access to either HPC or with the infrastructure provided by Galaxy. And I encourage you to read about the efforts we did to benchmark some of these pipelines at the Biohackathon last year and follow our Workflow Hub space where we'll be publishing our workflows in the future. So in terms of what we would like to see to make an annotation fully fair, we would like primarily a file format consistent with the software that we use. This is typically GFF3 or GTF, and accompanied with this, a full documentation of the pipeline, software and version, the data and databases used to annotate such a genome, and the genome version itself. We hope that with all of this information, we'll lose this need to duplicate efforts and computational cost of re-annotating genomes many times whenever a research group wants to analyze a genome. Finally, I would encourage you to get involved with ERGA if you would like to discuss more with the community about anything involved in the production of a reference genome, from sampling to the bioinformatics to even ethics and policy. You can register on the link here or follow us on any social media. Thank you very much.
Okay, great. That is the last of the lightning talks. I'll stop sharing my screen now. So Kim, your comment there, I was just about to say, the slides for all of these, the lightning talk presentations are available in the collaborative notes, I've put a link to the shared slide deck, which I'll actually paste directly into the chat now. 1 second, let's make sure I have the right link. I do. 1 second. And I'll copy and paste the link to the collaborative notes as well, just so everyone has them. So finally, you did mention. So we did mention that we possibly have just a chance here to catch up on any questions that we might have on the lightning talks before we move on to a discussion of the case statement or the work plan and the group's aims more generally. There is a question from Joshua in the chat. I don't know if you've seen that.
Yeah, so I saw that. Would you mind to elaborate on that, or should I take it as it's written?
Yeah, I guess at the moment what I'm finding trouble with is having flexibility with working with common data models. There's some platforms that require data models and maybe just completely a JSON format rather than as a SQL schema. And I was wondering if this tool can actually help a bit more with actually wrangling those common data models and could actually provide some tooling to slice and dice them a little bit to tailor them to your own needs.
So that might very well be. So the actual common data models, or I know these standards that you're mentioning, they're not supported directly, but the idea is for people to be able to add support for different models to this. And this is very much more focused now on the core functionality that allows that. It is still under development. But yes, the way you describe it, I think it should be a possible solution for this, specifically for converting the different various ways of expressing the same data model from JSON to tabular to databases to whatnot. That would be a use case that we are looking for at least.
Is it publicly available on GitHub yet?
Yes, it's on GitHub, but it's documentation is still a bit poor, but we can please get in touch, and that should be improved pretty soon, I think.
Okay, cool.
But there's also some examples available on different repo.
So sorry, you said that it's able to provide a way for people to actually use this to make potentially some transformation type things for common data models. So, you know, if this is something that I'm particularly interested in doing, is that something that I can use this platform to make available for other people as well?
Sure. So that's the idea. So basically, this is very modular. So you should be able to add different modules focusing on different types of data or different integrations, and make that available as different GitHub repos. That's the plan. So this part is not fully developed, but the main functionality is they're supporting all of this.
Oh, awesome, cool. Thanks for that.
Thanks, Joshua. I see Jeff has his hand up. Please feel free to ask a question.
Yeah, hi there. Hi, Sven. Good to see you again.
Right.
I just wanted to have more of a comment, and I think you know, Thomas's talk there around ERGA resonates very highly with what we hear here in Australia. So I think we've got a lot of researchers who have been frustrated by the fact that the global databases, you know, the annotation is mixed up with the assembly. So separating those into two and having the capacity for other, you know, the annotations to be shared independently of the assembly is really important, because often, as far as I understand, individuals will do a very high quality annotation. They'll go in and often manually curate certain areas to a high level, but when the data is submitted to the repository, pretty much the annotation is redone according to the standard pipeline that's used by that repository. So, I guess there's pros and cons for both methodologies, and, parts of the standard annotation pipeline may be better than the one that's been done locally, but other parts will be, better in the other one. So I think that to me really drives home the need for what the working group is trying to do. I guess a sort of next level, or something else that I think will be interesting, is, not all the annotation parts and annotations are going to be equal. And, how do we maybe reflect on quality of different parts of an annotation file? When someone's manually curated it, for instance, they've spent a lot of effort and time, making sure that part is of high quality. So I'm not sure if that's yet part of the, the plan of the working group, but I think that would be something, ensuring that can be associated with the file format or the interchange format is probably going to be really useful.
Yes, I agree with everything you're saying. So I'm not so heavily involved in the different ways of looking at quality, but I know that there are different standards, ways of doing that, and some scores, and this is work that ERGA is heavily involved with, and we would like to collaborate with them on taking aboard the metadata that's needed for quality assurance also. I think that's the level of detail I can say on that at the present moment, but hopefully we will be able to support that. I can also mention that, as Thomas said, that the different repositories are working on separating this annotation with assembly, and I don't know when they will be finished, because they've been working for that for a while, I think. But in any case, what we are suggesting is a more of an alternative community-driven infrastructure that can work on the side of this and can be more agile to users' needs possibly, and be a bit more on the cutting edge possibly of things that can potentially be adopted by the repositories later. That's at least a vision.
Lindsay, please go ahead. You have your hands up. I'm not, Lindsay, if you are speaking, I can't hear you at the moment. I don't know if anyone else is hearing any sound coming through. No.
Hearing anything?
If you're, Lindsay, if you're having issues with your microphone or anything, please feel free to post a comment or question in the chat, or yeah, unfortunately, we can't make out any sound at the moment.
So there will be a discussion also after the next slide set, so we can perhaps come back to that once you figure out what the technical issue was.
Anu.
Yeah, hi. I have a question about the OmniPy. Is that like a generic Python library, or is it specific for genomic annotations?
No, this is very generic. So this could be used across disciplines, really. So we're developing it for our needs here, but it's designed to be used for any data or metadata wrangling purpose, really.
I see. Okay, thank you.
You're welcome. So Lindsay, should we see if we can get sound
Yeah,
across
Lindsay,
now?
please go ahead.
Can you hear me now?
Yes, we can hear you.
Oh, okay, great. It was the headphone thing. Sorry about that. So yeah, so I'm one of the co-chairs of the multi-omics metadata harmonization group. So paying attention, would really love to coordinate, make sure that we capture some of the great work that will go on here. As a separate aside, I have a separate question about some of the things that maybe you will or won't tackle in this working group, and that is persistent citation of genomes and genomic references and papers. I know that's something that's really challenging. Either one, it's not cited at all. Accessions are somewhat persistent, but sometimes very much not. So that's something I'm very interested in as well, to see if that's something that I know the FAIR headers kind of touches on that a bit, bioheaders. So I'm just kind of curious if that's an area of standardization or recommendation that might be included in this effort as well. So, like how do you extract unstructured text from literature when it's not properly cited, or if it doesn't have a persistent URI? Is that something that you guys have discussed at all?
So if in terms of the latter, to extract text from literature, I think that will be out of scope, but I think within scope would be to make sure that the metadata that we are providing, or in the standard, that it contains useful identifiers and ways that it can be cited, can be attached to publications for easier access. So one of the ideas we had in the FAIRtracks project was at least to be able to extract metadata for a particular set of data, if you use some data for analysis, and then be able to provide that as part of the publication for reuse or for others to redo the analysis, for instance. I don't know if that answered your question, but we're very interested in any inputs on this, and I'm not on top of all of the needs on the journal side myself, so that's at least something that we would like to get more knowledge about.
Yeah, it's usually just a pain point in aggregating reported results. Sometimes it doesn't actually have a reference citation with it or something like that, and that's always a challenge. Another thing I was kind of curious about is linking across persistent identifiers, right? So you could have like an accession from one database to another, and it kind of touches on the point that Jeff made also, is like, how would you link assembly accessions to whole genome accessions to maybe like downstream annotation curations, maybe that sit in UniProt or something like that stems from a genome. So in terms of like a use case like that would be really interesting to kind of hear about what the thoughts would be from that.
Yeah, so that's very much within scope. So the source data that's used and the pipelines and all of that is really things that we should have in the model. So I think we should move on to the next slides, Ryan, and have some time for further discussion after that. So that will be say more about the plans of the working group and what we are actually trying to achieve here. And that's me, I guess. So I can find my slide deck again and share my screen. This here, and here, okay. So you can see and hear me now?
Yeah, all good.
All good, okay. So overview of the work plan. So just a bit of an introduction to this, just showing the history here. So this is one of the slides that I made in 2010 or thereabouts in the context of the analysis, and this is looking at the matrix of track hyperspace, meaning that if you have different reference genomes in one axis and different tracks in another axis, and then there's all these different axes that you can add with different cell types and genomes and whatnot, creating this sort of hyperspace. So that was an idea we had for analysis of this. And last year I saw this very, very similar slide in the presentation in the J4DH plenary, still trying to make this concept work, and that's very AI focused work that was very, very impressive, looking at different relationships between this. And obviously metadata is key to getting this sort of analysis available to get the collect the metadata or collect the data based on metadata. So that's an introduction to the use cases that we have defined, and this is the first use case. Discover and collect data sets to build AI models. That's the sort of headline of there. More detailed discovery of genomic annotations through harmonized metadata to improve the availability and scope of data sets for analytical applications, including methodology-oriented software tools and AI/ML efforts. That's the way we have described it. So focus on discovery of data and also collection or the accessing data. And this is focused on more on biomedical analysis focus. So the next use case is then the biodiversity use case, which we call genome annotations in biodiversity as FAIR objects. And in detail, the way we have written this is that we wanted to establish genome annotations for biodiversity assemblies as FAIR objects with improved metadata and integration with relevant infrastructure and tools, including both manual and automated annotation of genome assemblies. So this is very much answering to the need that Thomas and Jeff and others have said something about to be able to work with annotations independently of assemblies. And this also answers to some needs written in the report from the Earth Biogenome Project last year, in that there is a need for metadata for annotations and standards for that. So that's the sort of two main use cases, and then there's a third use case which is more focused on data accessibility or interoperability. There's this solution called Track Hubs, which is a way that some existing genome browsers have been sharing data. Basically, Track Hub is then a collection of track of data that you can set up yourself to share the data for visualization and analysis elsewhere. And we have been previously worked with the Track Hub registry, which is an EMBL EBI solution for registering different track hubs. So we want to enhance genome browser related infrastructure for track hubs with harmonized metadata and related services to simplify the process of generating, hosting, and registering metadata-enriched track hubs. So basically adding metadata, standardized metadata to track hubs, that's the main goal here. Okay, so how we have set up the working group plan. So we plan to have 4 different main deliverables of the working group. The first of which has been mentioned several times is a recommendations for a minimal FAIR metadata schema. So here we want to harmonize efforts from different projects, including the Gecko data model and FAIRtext model, both which was presented here. And also there's the IHEC, which is International Human Epigenomics Consortium, where we have a member from the data portal, working on their portal there. So that's another data model, and then also different data models out there. So trying to harmonize those and add the needs of the different use cases for creating models. That's the first deliverable. And then there's a set of core infrastructure recommendations that we will look at to be able to help with the sharing of data according to metadata. the first of which, as also been touched upon, is the focus on executable metadata transformation pipelines. So basically, you have metadata available in different projects in different contexts, and you want to turn that metadata, transform it into this harmonized, according to this harmonized model. Once we have that, and then there's a number of different data wrangling and cleanup steps and things that we need to do, and you want to make this as general as possible and scalable so that you can automate it and run these pipelines when there's new data, for instance, or when the model changes or when the ontology changes and all of that. So this is one of the things that we'll try to look into. Second part is recommendations for persistent storage. So basically, we would like to provide a way for this harmless metadata to be stored somewhere so that it can be discovered by different services. There's also a need for identifiers here, possibly content derived. So just a bit side note here, this is from a related work in the Global Alliance for Genomics and Health, a new standard which is coming now called sequence collections that can basically identify a reference, uniquely identify a reference genome, and also genome browser instances. So Nathan, co-chair, has been leading this work, and I'm a part of that. So we very much want to adopt this into the model. And a related question is if it's possible. So this is content derived, providing an actual genome sequence, you will be able to calculate the identifier. And is that a option that can be used also for annotations? That's a question that we will look into. And the last part is a standardized API for search. So we're not providing the actual search engines and so on here, but a way to standardize this so that the different people can plug in and still be able to provide a standardized API for downstream services that want to get access to this discovery capabilities. So with these 4 different elements, we think we will be able to create a community-oriented infrastructure that can be adopted by different tools and services. And then there's a proof of concept integration with third-party services. So that will be done a bit outside the actual working group, but the different projects involved here trying to adopt these standards into their solutions. And lastly, resources that support community building and adoption. So we want to build a sustainable community with people with a passion for data integration. And we really want to have different kinds of people involved here in the group. So from data producers, people from the FAIR RDA community, or RDM community should be here, I guess. Tool developers, domain experts, and also the analytical end users of these solutions. So everybody and other people that's not part of this. Yeah. And then collaboration, adoption of other recommendations. So here's a list of different working groups or interest groups that we have thinking perhaps is of relevance. The omics group is not part of here, but it should be there definitely. So that's I forgot that one. Not only in the RDA, but also in the GA40H. So there's a crossover possibility here, and that's, I think, also important. There is things being done in different contexts that are very overlapping, and getting more people involved in both, I think, is a strength. Yeah, so that was what I had for the working group, and then we can go back to the discussion. So list some focus areas here, but we can really discuss anything. But the focus areas that's listed was any challenges with the use cases, any individual tasks from the work plan, intersections with other RDA groups and efforts elsewhere, or whatever you would like to comment on.
So yeah, so please feel free, everybody, to come forward with any questions you might have. So obviously, Lindsay, you did mention the multi-omics group, the Oracle for Research group. So yeah, absolutely, that'll be one of the ones that we will list there. There's a question in the chat from Kim. I guess was on this more focused on the slide about the kind of building the community, asking are we interested in including repository managers, so the INSDC contact points?
Definitely. So we have some contact with the ENA, trying to get one of their people aboard the working group, but we have not so much contact from the NCBI side. So that's definitely of importance. So if you have some somebody on the NCBI side that would like to work with us, then that would be highly appreciated.
Yeah, I'll have to see if people have, this is Kim speaking, I'll have to see, what people's time availability is. I find these presentations really interesting because the INSTC is also doing some introspection and future planning right now.
Yeah, just to mention a contact point, that's basically what you asked about, and that's at least a very good start. So if you can provide a contact point or be one, then we would be appreciative of that.
And Lindsay, I see you've added something in the chat, but please, you have your hand up, so please feel free to unmute.
Yeah, have you thought about also just linking up with some of the initiatives that have developed a lot of genomic standards? Sometimes I kind of wonder if there are elements that can be adopted from this working group's effort into existing like reporting guidelines. So down to, how you link and include certain accessional information for genome annotations as well. I know that they're pretty open to adopting and integrating new things. You have like GSC, you have a few other alliances that you already mentioned too. It might not be a bad idea just to see if you guys can wrangle someone in from there and see if they're also interested in adopting or presenting some use cases as well.
So that's definitely a very important. I didn't list them there. But what I did list was in the GA40H. There's a working group called Experiment Metadata, which we are very much want to collaborate with, and they also are collaborating with the GSC, and they will actually have a hackathon on that in the next Genomic Standards Consortium meeting in August or when it is. So there's a link there already, but strengthening that link would be also a very good idea. Yeah.
Yeah, I think Tim, our co-chair, also is going to be at that meeting. So maybe if you guys have some folks there too, I'll pass that on to him.
Sure. Can you write that name? I didn't catch it.
So I think that's one of the co-chairs, Lindsay, from the Tim van der Bosch. I'll put his name in there. Just I'm trying to keep notes here at the bottom, but yeah, it's fine. I have a note of the co-chair.
Thanks. So now it's the moment of silence. Yeah, so I guess, I mean, one thing that I was also going to mention, I have a slide on that bit later, is that all of the 4 co-chairs that we have now, they are mostly on the biomedical side and not so much on biodiversity. So we are looking for a co-chair that has biodiversity as more of a focus in order to drive that part of work. I think it's a very important and highly needed use case. So so that's still a bit of a search, but we are starting anyway, and we have some connections into that community. But if you do know of someone that might be a perfect candidate for that position, please tell them or tell us or something.
I'll just say that I don't have any clear candidates at the moment, but the biodiversity space is an area we are expanding into. So I will talk to people and see how that all pans out. But that's a priority area for us. So I'll be in touch.
That's awesome. Thanks, Jeff.
So this is Anu again. In terms of external groups that you're interacting with, beyond J4GH, have you been in touch with anybody else? I mean, and the repositories, right? Like what Kim mentioned.
Not so much. I were in contact with some groups in Australia because I visited there with the help of Jeff last year, but that's a bit just informal. So if you have some suggestions or connections that you think is relevant, then please let us know, or we're very interested in that.
No, I don't have anybody in mind, but I was just wondering. Thanks.
Yeah, there's also, I mean, the people, co-chairs have been involved in different projects previously that's been closed and so on. So what we are lacking, I think, is more of the major browsers, and specifically the UCC genome browser, because they have not really been involved. We have contact with Ensemble, which are the two main genome browsers. So if there's any connections to them, they are informed about this, but they have not been engaging much. So that is at least a link that we would like to open up. Yeah, that's actually the question. Yes, so there's been some emails sent to people, but not so much open communication yet. That happened earlier in the FAIRTrax project through a person who's now quit academia. So that link is a bit gone. Yes, so Kim writes that the GSC interests also seem to overlap to this working group to some degree, and I definitely agree. I think the goals are a bit different though, because the GSC is mainly focusing on storing any metadata, or the full level of experiment metadata, and what you should store for the future, while we are working more on a sort of minimal use case for discovery and for the sort of main use cases, and then linking back to the full metadata. That is the model, at least we were using in the FAIRtracks project. So it's a bit different scope, but definitely overlaps.
We are getting close to the top of the hour. So I know we're not like exactly setting people homework or anything, but there's one or two more slides that would be worth covering, I think. So I know maybe unless there are any further sort of questions or comments. I think the main thing, what I will do, and I'll post this in the chat right now, is to encourage people really to join the working group on the RDA website. So I've just posted a link to the group there, and if you're signed into to your profile on the RDA website, we encourage you to just hit the green button there on the right hand side of that page, and to join the group. Obviously, it's the same with all RDA groups. You're free to sort of join, and if you're just to be kept up to date with what's happening in the group, then that's obviously where more people that are joined, the better. There's no join the group. There's no sort of requirements for you to contribute to meetings or join or anything. But obviously, the more effort we get, the better the better the group will progress. But yeah, just even if it's the case you want to just be kept up to date with the group's efforts, please encourage you to hit join and maybe so yeah, just sign up to the group's mailing list. I guess people.
Are you sharing the last slides, or should I? Or
I'll do it here, so I have them on my screen.
Yeah.
Cool. So I should be looking at the slideshow now. Yeah, so just in terms of the next steps. So first thing before, like in terms of the next steps, yeah, we as Sveinung said, we've had the case statement is awaiting so we've gone through the whole community review process and received some very positive feedback, and just a few minor revisions as we noted here. Hoping to expect, so we've resubmitted that and hoping to hear back from the RDA Council soon, maybe even by the time we have the repeat session for this group, which is taking place next Thursday at 14 UTC. So yeah, so that time mightn't be suitable for some of the people who've joined us today, but if you do have colleagues or others in the who are attending the plenary, please point them in our direction for the session next Thursday to join. I'll put a link to the case statement as well in the collaborative notes if you want to read the full sort of document, and it goes into a bit more detail on what the group's aims are. As I've said, please encourage you to join the working group as well. And if you again, just please pass that to any of your colleagues who might be interested in keeping up to date with the group's activities, or to actively contribute. That will be even more appreciated. As well as this, so we'll be setting up. So the group is close, and we're just waiting to hear back from the RDA Council. So when we are officially endorsed by the council, we will be setting up the regular working group meetings to sort of get the ball rolling really with the group's activities. So we're planning on holding these on the third Tuesday of every month. So the first meeting will be in just about a month, just over a month from now. So planning on 14 UTC for now, but obviously that will we can discuss that further if there's depending on the kind of makeup of the groups in terms of geographical diversity. But please, we'll encourage you to, if you'd like to join us at these initial meetings, please, as I said, please feel to join the group on the website, and we'll be able to get in contact with you that way.
I think you had another slide.
Yeah, there's a final slide. Yeah, that's right. So yeah, so as Sveinung said, we're still looking for a co-chair with a biodiversity focus. Yeah, Sveinung, I'll let you speak here.
Yeah, no, so I think I said all of that. Just the last list of people that would like to thank here. So Ryan and others from the RDA Tiger team has been very, very helpful in setting this work group up and all practical and support needs that I have had. So that has been very great. Ari Asmi has also helped out personally and also in terms of getting us connected to the RDA Tiger proposal. Peter McCallum, Felix here, put us in contact with Ari Asmi, and also Jeff, who is here. Thanks a lot for the help that you provided, especially in my trip to Australia last year and all the things. So thank you all.
So yeah, we're just hit the top of the hour. I guess it's getting late for it's getting extra late for Swineham. It's getting late for me as well. Might be getting early for some people, might be mid-morning. But thanks everybody for joining. Appreciate it. As I said, we'll be holding a repeat session next week. So if you do want to rejoin us, some of the other co-chairs will be joining us on the session on the day as well. So we'll be able to you'll be able to put any questions you might have. You might think of in the meantime, you'll be able to put it to them directly next week. But again, if you have any colleagues who might be interested in the group's activities, please point them in our direction. And yeah, thanks again for joining. I appreciate all the contributions. And yeah, I will bring the meeting to a close now, I guess.
Thank
Thanks a lot.
you. Bye.
Everybody.
Goodbye.
Thanks. Bye.